Thank you so much for that kind introduction, and thank you so much for having me here. It is such an honor to be in this room. And can I ask if you feel comfortable? Will all the survivors stand up? Do you feel comfortable? Oh my gosh, this is amazing. Let's give a round of applause. And I just wanted to say that you all are why I get up and go to work every day, and I'm passionate and so excited. Lung cancer biomarker testing is really my passion. It's what brought me into my medical specialty, and I'm really, really excited to be here and talk to you guys this evening. So thank you for having me. And thank you for those that were able to join us on our laboratory tour this afternoon at Foundation Medicine. I hope you were able to get a peek at kind of what goes inside the biomarker and genomic profiling process and how passionate and dedicated all of our staff are at Foundation Medicine. So I'm really glad that some of you were able to join. So I wanted to start off talking a little bit about my field, molecular pathology, and what we do at Foundation Medicine, and just a little bit about my journey and why I'm so excited to be in this field and where we are today, and kind of some of what just Emily was talking about. Like, this is just such a great time of innovation and new opportunities for our patients. And then I'll end with a little bit of time for questions as well. So I mentioned this before, I'm a molecular pathologist. I don't think many people, including some of my physician colleagues, really know what that means. So I did an anatomic pathology residency training program, and I think most people, when they hear pathology, they think CSI on TV. So it's not nearly as fun and exciting as that. But what pathology does is a lot of microscope work. So when you got a biopsy, if you had surgery, if you have a blood test, somebody has to look at that tissue in a microscope and get that diagnosis. So that's kind of part of anatomic pathology. And then there's this whole other field that's laboratory medicine, and that's running laboratories like ours at Foundation Medicine that does all different kinds of testing. And so I started, and how I kind of ended up where I am today, and I'm so passionate about the field, kind of starts when I started my anatomic pathology residency program in 2013. And does anybody know what the FDA approved in 2013? Maybe some of my AstraZeneca biopharma colleagues. So this was the year that the FDA approved erlotinib for patients in the first line setting that had advanced non-small cell lung cancer that had EGFR mutations. So erlotinib had actually been on the market in the second line setting since 2004, but it wasn't until 2013 that it gave approval associated with the biomarker. And it was actually discovered after the first clinical trials in the post hoc exploratory analysis, where they were like, wow, you know what? We got this drug. It works in some patients with lung cancer. It tends to be, there's a higher response rate in patients with Asian ethnicity, higher response rate in non-smokers. And it was after the fact that they realized that, oh, hey, the patients that have these certain mutations in EGFR, they respond really well. And it was just kind of amazing. And so I remember just hearing this story as a first-year resident in my medical training, thinking that this is just amazing. We like to think of a lot of medicine as being very evidence-based and based on science, but I think all of you who are very experienced in the healthcare field know there's a lot of trial and error. There's a lot of art to medicine. And here was a discovery that, hey, we can identify the exact patients that are going to respond to this drug. We don't need to try this chemotherapy. We don't need to do this. We can identify who's going to work. And that was just amazing to me. I had done graduate school before and had worked in the field of lupus genetics. So I had a genetics background. I was like, this is what I want to do. This is the future of oncology. This is amazing. And this is something I want to be able to work and do research and innovate and offer this kind of testing to patients. And so that's what I decided to do. And it was really an exciting time throughout my residency and fellowship. This is kind of when, in the laboratory that I was at the hospital, I was at Dana-Farber Cancer Institute at the time, I got to work on the first test for EGFR, for KRAS, and for ALK. At the time, we didn't have KRAS targeted therapy, but it was very helpful to know if you had KRAS, you didn't have the other ones that had targeted therapy. So it was kind of an easy way to get the right result. And by the time I finished training, we weren't just looking at 3 different tests and genes. We were sequencing hundreds of genes. We were looking at complex biomarkers, not just for targeted therapy, but things like tumor mutational burden and PD-L1 and microsatellite instability for immunotherapy. And this was from when I started residency to when I finished fellowship, before I'd even started a faculty job. So it was just amazing, just the explosion in the field, and just really, really, inspiring and really exciting. And it was just a whole new field in oncology, right? Now there was targeted therapy. That was just, not a thing more than 10 years ago. And now there's immunotherapy. And so, it's just amazing what we've come in the past 10 years. And so, I like to think about 60%, kind of depending on the histotype of lung cancer, patients have a targeted therapy, which is a lot. If you're looking at adenocarcinoma, it's even more. And what we know is patients that have genomic profiling and they have a targeted alteration, if they get therapy, they have better quality of life, they have better outcomes, they have better survival. Unfortunately, what do we know? There's data, the data's a little old now, but there's electronic health records data from community-based hospital systems from 2018 to 2020 that less than 50% of patients receive the 5 most commonly recommended biomarkers from guidelines. Less than 50% of patients. That's just really heartbreaking. I know one of my colleagues at AstraZeneca and I were just talking about the challenges in our healthcare infrastructure and setting of being able to get this testing. I think there's a lot of multifactorial reasons why it can be really challenging for patients to get testing. Some of it's access, some of it at some point in time was payer coverage and reimbursement. Some of it is lack of education upon providers, which is hard to imagine. And so, that's just really, really critical. I'm thinking about, okay, we know what we can do to really improve survival and outcomes in patients, but it's not getting to everyone. And why isn't it getting to everyone? And so that's why I think organizations like LCI and other advocacy organizations are just really critical and really amazing and really inspiring, because I think this is really key, and why we have so many survivors here in this room, because people here were able to get, on the right therapy and have the right guidance. And that's something that we can really help to advocate, to encourage friends, family members, work with advocacy groups, just really kind of what the importance of getting genomic profiling and getting biomarker testing done. Even if it's difficult, maybe sometimes it's worth it. Sometimes there's not enough tissue. Sometimes you have to have a repeat biopsy. Luckily, in lung cancer, those patients tend to have, we were just having a conversation earlier, lung cancer tends to be one of the tumor types that tends to be in the circulation more often. So sometimes liquid biopsy is a really great option for lung cancer. So there's a lot of different options that we can do. And those are some of the things at Foundation Medicine that we do genomic profiling. We do it on tissue. So whether you have a biopsy, you have an intrabronchial ultrasound biopsy, you have a metastatic biopsy, or if you have surgery, these are specimens that we can test the tissue, and we can see, okay, what alterations are present in that tissue, so we can guide therapy for that patient. Not how do we treat this disease of a population of patients, but how can I help this patient? How am I going to give them what's going to give them the best chance? And so that's something that we do. We do it looking at hundreds of genes at a time. Again, very different than when I started in the field over 10 years ago. And then now we can also look at what we call liquid biopsy, which is basically just a blood draw. We can see what parts of the tumor can we see in that, and that's really phenomenal. One, if you don't have enough tumor tissue, right? If you just didn't have enough and there wasn't enough for sequencing, or perhaps you've been on therapy, you're doing well, but maybe there's evidence of progression. So it's a really great way to follow up on patients longitudinally, because it's very easy to get a blood draw, much easier than getting some more tissue biopsy. So it's a great way to follow patients. You can look for things like, is the amount of tumor DNA in the circulation going up? So that's a sign that maybe the patient's progressing. You can also look for resistance mutations. Now there's a lot of creativity with patients that develop resistance mutations of combining different targeted therapies. So targeting the original alteration and the resistance mechanism. So this is another great way that we can monitor patients over time and think about how we can combat any mechanisms of resistance that are coming up. And a liquid biopsy test is a great way to do that. Another advantage of these liquid biopsy tests is that if you're taking a biopsy, you're sequencing it and looking for what's in the tumor, you're only looking at what's in that tiny little piece of tumor. And oftentimes our patients might have disease all over the body. So what's really great about liquid tests is that's kind of more of a holistic picture of what's going on in all the different aspects of the tumor that a patient might have if they have multiple sites of disease. So that's another great benefit, especially in the setting of on treatment and perhaps some developing resistance. So those are some exciting tests that we have now. We're working on, and again, why I'm really excited to be doing this, I'm going to say, what are you excited about? What kind of innovations are you looking at? So looking at things like early cancer detection, that's something that is really exciting that our company is working on. Especially I think for the general population as a whole, but it's a really great option for patients that are high risk, like you mentioned. There's different right now CT scanning protocols, but maybe we can get more sensitive, right? Maybe we can detect it earlier, sooner than you could see on a CT scan. So I think that's very exciting. That's something that we're working on at Foundation Medicine. And then even more sensitive monitoring tests in minimal residual disease tests, so that I was just talking to Emily, and I think you were saying you were getting scans every 3 months, maybe, right? And so maybe we can do a blood draw that you could go into the clinic and get a blood drawn. Maybe we can detect something in 4 weeks instead of 3 months. Maybe we can catch it sooner. And so that's something, because we're working on developing tests that are very, very sensitive, where you can detect maybe 1 out of 1 million in your blood. So I think that's really exciting advancement too. And maybe we can do better than looking at scans every 3 months. So those are some exciting innovations and new test developments that we're working on at Foundation Medicine. There's obviously a lot of other companies that are doing kind of similar work to us. Another thing that I'm very proud of, and one of the reasons I'm proud to work at Foundation Medicine, is because one of our really main goals and objectives is access. And access for patients, and be able to get this test and not have a bill for several $1,000s, right? So we've worked really hard and been able to have Medicare coverage for both our tissue-based and our liquid-based tests, which I think has really opened up access to a lot of different patients that might not have been able to afford our tests. And it's really a main priority for us is to access and get payer coverage for our tests, which I think I'm really proud of. Because that 50%, less than 50% number isn't acceptable. So we're trying to do everything that we can to make it more accessible to individuals. But really, I think we still have a long way to go. And I think advocacy is really important. So that's why I'm very, very grateful to all the work that you do at LCI. This is my first time here. I hope I get to come back and learn more about the organization and participate, because like I said, it's my passion is biomarker testing in lung cancer. And I've just been kind of really amazed at what's happened in the past 10 years. And I think we can continue to advance both in technologies and cool innovations, but both in kind of more grassroots advocacy and outreach. So